I'll turn it. Okay, cool. All right, welcome everybody to Molecular Evolution 3. My name is Donovan, German. I'm going to be the chair of this session. I just found that out. I'm at UC Irvine in Southern California. I'm your first speaker too. So I'm going to talk to you about convergent evolution of herbivory following different trajectories in prickleback fishes, which are these guys. They're intertidal fish native to the Pacific. So a lot of the questions in my lab stem from specialization, trying to understand how different organisms specialize in different diets. And we study things from the isotopic level up to the community level, with a lot of stops in between and a lot of focus on what's going on in the gut, what's going on with digestive enzymes, what's going on with gut structure and function. And so this talk will mainly be focused on enzymes, the chemical mechanisms by which organisms digest their food, and these fish digest their food. So when you look at these different diets between herbivores and carnivores, for example, they're very different biochemically. Right? So there's a lot of carbs in a herbivorous diet, a lot of protein and a lot of lipid in a carnivorous diet. So very different biochemical constituents. And this part is the part I want to focus on because the algae that the fish that I study eat are loaded with starch. About 10% of their mass is starch. And so the enzyme that breaks that down is amylase. And across organisms, not just fish, I'm going to show a few fish examples, this is where there's clearly been a lot of selection on the gut in terms of enzyme activity levels. And this is an example of silversides, another marine fish. That's the amylase activity on the y-axis, different taxa here on the x. And you can see that this herbivorous population of Atheronops affinis actually has elevated amylase activity compared to carnivorous populations of different species, or even its own species. Okay, so this is an estuary where it's herbivorous, kelp forest where it's carnivorous. So same species showing major differences in their enzymatic activity levels. Now if you look at this in a phylogenetic context, this is looking at minnows in the southeastern United States. You can see that there's elevated amylase activities in the herbivores here in the genus Campostoma, and mostly lower activities in the carnivorous genera Nokomis, except for a single population of Nokomis leptocephalus that's herbivorous. So there's this clear signal relating between diet and amylase activity. The more starch, the more amylase activity. And this isn't unique to fish. So it's clearly elevated in herbivores and omnivores, but not just in fish. It's in birds, bats, and us, and in canids. A great study looking at genome evolution between dogs and wolves, showing that was one of the clear areas of selection on amylase and glucose processing. When you look at some of the evidence, this is independent contrasts of amylase activity against percent dietary starch content in birds. There's a clear correlation there. And just to get to that primate example, Perry et al. 2007 have this great example of gene copy number variation in salivary amylase in humans. So you can see this Japanese individual up here has 14 copies of amylase in their genome. A Bianca individual doesn't eat much starch. They have 6. And you go down to our most common living ancestor, the chimp, they only have 2. They eat very little starch. So a correlation between gene copy number and the amount of starch in their diet. And of course that relates directly in this case to enzymatic activity level as well. So moving on to what I want to do is I'm very interested in this question, is what are the genetic mechanisms underlying elevated amylase activity levels? Is it always about copy number? Okay? As it's been, at least in humans, and in the dog example, they also have elevated gene copy number for pancreatic amylase. So is it always about copy number? And what are the functional and fitness consequences? So we're going to spend some time talking about the functional consequences today. I don't have much on the fitness just yet. So because I think it's important to look at the animals we study, not just get focused on them as a bag of genes, I think we should at least see who they are. This is Semidictes violaceus, an herbivore. This is Xiphister mucosus, another herbivore in the same family. Xiphister atropiparus is an omnivore, and Anaplarcus purpurescens is a carnivore. And there's one more. If I take these chirus, that's also an omnivore that we get in Washington. And these fish are actually all sympatric. You can literally catch them under the same rock, on the same site. So they have different diets, but they're living in the same general habitat. And so we made a tree here, a 3-gene tree using Site B, 16S, and TMO4C4. This is now published, Kim et al. 2014. And it was very clear from this analysis that Sebedictes and Xiphister, the two herbivores we're interested in, are not in the same clade. They're in different parts of the family. And indeed, we have an entire group here where omnivory and herbivorous popped up as the Xiphister clade. And down here, Anaplarcus purpurescens is in this largely carnivorous clade. And in general, carnivory is the basal mode of the family. Okay. So we're going to look at not only our herbivores, everybody who's got asterisk, but we're going to include this carnivore as well as one carnivore from Japan, Dictyostoma bergeri, that's in the same tree here with Sebedictes violaceus. We're going to compare their amylase genetics and biochemistry. And just to prove that amylase activity does correlate with diet in these fish, this is pancreatic amylase activity. Their pancreas is part of the pyloric cecal region in the gut, so it's very easy to extract just that. And so we have amylase activity of different taxa, and you can see the herbivores and this omnivore at least have elevated amylase activities compared to this carnivore and this omnivore, which eats considerably more animal material. So the correlation is here. All right, so we're going to look at amylase genetics and biochemistry. So I'll start by looking at the amylase sequences, and I'm going to look at the amino acid sequences deduced from the cDNA sequences. So we'll start there. A little light reading for you. Take your time. I'll point out 3 important things, or 4 important things. First, we have, you can see there's 2 sequences for Sebedictes at the very top, 2A and 2B. We consistently got 2 separate sequences for that species. And we have single sequences for everybody else. Some other important facts are that the active sites, my boxes got moved over, are all conserved, which should make sense because actually they're conserved across vertebrates for amylase, as are the calcium binding sites. These are all conserved across our taxa. But what we see are several different areas where Sebedictes has unique differences that result in amino acid changes. And that's just looking at Sebedictes against the other taxa. There's also incredible differences amongst those 2 variants that Sebedictes makes itself. So we're going to zoom in then to look at what's going on with the diversity of amylases in Sebedictes. And so if you look at the 10 amino acid differences between those 2 sequences that they have, we have 2 main forms, 2A and 2B. So this is just focused in on those 10 amino acid variants. So we get these over and over and over again when we clone them. However, we also get other variants. We get this, 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 this. We also get this, this, and this. We're getting about 10 or more copies or variants I think we're looking at. And some of these is pretty obvious. For example, right here, where there's been crossing over, there's been exchange of material across the different genes. So we find that of interest here in terms of being like alleles that have crossing over among them. One other aspect is we look at the 5' untranslated region, and Sebedictes has 2 separate ones of these. The Xiphister mycosis, the herbivore only has 1, as do all the other taxa. So a lot of this is matching up that they have at least 2 main forms, 2A and 2B, with variants, subvariants of those. Now, what about overall gene copy number and expression levels? So we did gene copy number using gene dosage curves, so actually just using a known amount of the copy of the gene using RT-qPCR, and then to determine how many gene copies our fish have. And you can see here's the diploid gene copy number for different species, and we have 4 or 5 for most of these taxa. But for Sebedictes, whether we use a general primer that grabs both, that gives us about 12. But when we use individual primers that are unique for 2B or 2A, we get about 6 and 6. So they appear to have about 12 copies, about 6 of each of this 2A and 2B form. So there's clearly gene copy elevation here, but not here, relatively speaking. And in terms of just looking at qPCR expression levels, we can find both 2A and 2B are expressed relative to our LA control gene. And I have Xiphister on here as well. I'm not really comparing these to each other in terms of magnitude, given that these are wild caught fish. These are in the lab on the same diet, although they do eat similar things in the field. So the main thing here is we're definitely getting expression of both isoforms or variants in Sabidictes, and we get one in Xiphister. All right, what about looking now at the protein level? So we're going to look at electrophoresis and a protein model. So what we do here is called native page. So we actually run native proteins out on a gel, and then we actually stain for activity. So we actually put the gel in starch, you let it incubate, the starch will permeate the gel itself, and wherever the amylase is, it's going to chew up the starch, right? It's going to break that down. So then you stain for starch afterwards, so the gel turns brown or blue, and you get clear band areas where your amylase is. And so we have clear bands for 4 in the main taxa here. I'm not showing Anaplarchus on this one because they have such a different isoelectric point. We have to use different conditions, so it's in its own gel. And the main point here is that what's driving all these different bands is isoelectric point because their molecular weight is all very similar, but the isoelectric points vary a lot. So we have 7.86, 7.59, 7.73, and 7.32. So very different isoelectric points based on their different sequences. But what was going on was we still were only getting one band here. We know from the genetics that Sabidictes has 2 variants. They're very different. So we should be picking up the second one. So we reasoned if we go above the isoelectric points for all of these to where they would then be negatively charged, we then could actually just get our variant for 2b. And that's indeed what we found once we went to pH 8.2. We get 2b only for Sabidictes. We have, if I take these chires right here, they get nothing. We also concluded a porcine control and got nothing. So we have now protein evidence that they're both there, so expression and protein. All right, and then what we were quite interested in was how different, or what does this different variant mean for Sabidictes? And so we, with Brett Lockwood, we were able to model the Sabidictes amylase, and the differences amongst the 2A and 2B variants are in yellow. The calcium binding is in green, active site in blue, and there's a flexible loop here in red. So there's 2 main differences that we find are significant using SNAP analysis as well as in the model itself. The first one is aspartate to asparagine at position 174. This matters because of where it is. At position 173, we have a calcium binding site. At position 175, we have a cysteine forming a disulfide bridge with another cysteine at 156. So this position at 174, this change from aspartate to asparagine from a charged to a non-charged amino acid appears to really matter. And when you look at the model, you can see this is aspartate and this is asparagine. You actually lose a salt bridge with a lysine at 155. So this actually changes the flexibility of that region of the protein. And changes don't have to be in the active site to actually change the function of the protein, as we know. And here's another one that we haven't modeled extensively yet, but there's a change here from glutamate to glycine that's highly significant in terms of change of charge, polarity, and it actually may change the conformation. We're still investigating what that means. Okay, so we have 2 main variants. We're getting them expressed. And here we're suggesting that there should be some pretty wild differences between them. So what does this mean for the animal? So this is where we're going to actually look at activity level against different starches. This matters because, oh, I'm almost done. Red and green algae make different starches. Okay? So this makes amylose, this makes amylopectin. So very quickly, just to run through that, different starches, you can see there's actually really no difference between the 2 taxa. The taxa themselves hydrolyze different starches at different rates, but there's no real difference between Sabidictes and Xiphister. So Sabidictes has all this gene variation, Xiphister does not, yet Xiphister seems to hydrolyze starches just fine. And so just the take home here is that, what does it all mean? We have expression of more genes in Sabidictes, fewer genes in Xiphister, but they end up with the same general phenotype, elevated amylase activity, and being herbivores, right? And these guys also seem to be more reliant on microbes than these guys, but I don't have time to share that with you. So there's just a lot going on here, and my take home is there's more than one way to skin a cat in terms of actually becoming an herbivore. They don't always have to follow the same route. And with that, I'm going to be done, and I do not really have time for questions, and I apologize for